Having good collaborators in the first 2 years, crucial. I will recommend having a collaborators that could help you or give you some leeway. Take your little wins because it's hard and it takes time and it's a lot of effort. Whenever I have a mentor that allowing me to have my bubbly personality, then I realize like it's so much better because it's a career, so I have to do this as I love what I'm doing and not just doing it because we want to get a paper out. We love what we do. It's such a long run. If you have the opportunity to be happy and balance your life and not be all the time into the lab, by trying to incentivize that with my students.
Welcome to the Science with Impact podcast, where we help research scientists broaden their societal impact. Today, we have a wonderful guest named Dr. Valeria, who is going to walk us through her journey in STEM. So, Dr. Valeria, could you please introduce yourself?
I am Dr. Valeria de la Rosa-Reyes. I am a Puerto Rican anatomist, professor, and neuroscientist researcher in UPR, University of Puerto Rico at Bayamon. I teach undergraduate students and have a lab with undergraduates. And I'm very happy to be here and talk more about science and how we use it every day.
Why should our listeners, who are primarily research scientists and organizations looking to expand their societal impact, listen to our conversation?
I think everyone that is into science and education can learn a lot of different experiences. If you want to learn about how much resilient or hard work it's done in different institutions, and if you want to keep working up and be motivated in education, you listen to different stories and learn from different people, and just having the opportunities to learn and continue work around.
Could you tell them how you got to know me and Science with Impact?
I met this wonderful person, Dr. Vanessa Rosa, at the SAGNAS Leadership Institute, and it was a great experience. We were 20 early and mid-career professionals in different areas, and Vanessa is such a great spirit. She very driven and motivated, and it was wonderful.
I'm so glad that we're here together today. Can I ask you, what were some of the impactful moments in your life that led to you becoming a scientist?
I discovered that I love research when I was young or as an undergraduate. How I get into science or curiosity, basically, when I was with my grandma, the farm. She had a lot of animals, and we got a lot of land with nature. So it was really great to be able to explore and observe. I knew that I loved biology at that point, probably because everything you hear more is the medicine, and that is like the role models, especially back before social media was such an explosion. So I got into research when I was an undergrad. I got the opportunity to do one of these undergraduate enhancers of research. It was called the RISE program. It was funded by the NIH, and we have a mentor and a project that we can work on 20 hours a week. And I just started working with molecular biology, elucidating some proteins and how the roles work around using frogs at that point, and we were characterizing some proteins in a pathway. It was a lot of learning, and then I did some summer research internships, and I fell in love with zebrafish and other types of research, especially in the microscope. I decided to do the Ph.D, right, and fall into this big hole of years of doing research and learning a lot of anatomy and neuroscience and do a specific project. And then I discovered teaching as one of the passions that I do.
What do you love about teaching the most?
From teaching, it's when you see people faces and making connections and applying. I feel like one of the things that I aspire is that the students are able to explain to everyone around them or the communities, being leaders and actually learning or communicating the science and the basics. It was one of those moments when you have families that are older and they need a lot of more care. And when the grandkids are able to join in and make them feel more understanding of their bodies and how everything is working, and kind of going to the doctor feels less scary because you have someone else. So that's one of my goals. So the students, at least, even if they're not going to be medical or health professionals, they can understand enough about the body and the anatomy or the brain and kind of feel comfortable sharing how something is working or not.
I can see why that would be so fulfilling.
That's one of my favorites. And in the lab, I feel like when they kind of do something, even if it's just cutting a tissue and looking at the scope, I'm feeling like the ownership of this is my work that I put up. And then you can see the image and analyze some data, do a graph. So it was really fun to see them growing and how they are developing as leaders.
You're creating a new generation of scientists. That must feel so wonderful. As a first generation professor from Puerto Rico, what unique challenges have you faced in supporting your students as they transition from undergraduate to graduate school?
The University of Puerto Rico and my department has been developing a system that having a mentor that is older faculty that teaches you or have meetings every semester. So develop a plan and you work out some goals and what resources you may need for reaching them. So it's been doable, and all these networks that I have seen other collaborators and peers that are just great among all the US, and you can see how they've done it in different areas. Of course, in Puerto Rico, we have a few challenges. We are an island, it's kind of hard sometimes to get stuff in terms of the electricity grid. We have a few times that we need to have all these backups of generators and battery backups for all the labs and the fridges and all the more sensitive areas that can be impacted by the electricity shutdowns. I have wonderful collaborators that been great, allowing me to do some experiments in their spaces while I am gathering up or building my own space resources. Funding is always one of those struggle, and I think that's something that happens across US at this time.
What advice would you give to an early career researcher who wants to build a community of mentorship around them? For people like yourself, what has been most successful for you in building a community of mentorship so that you can do these cross lab investigations and support one another?
I will say if you're applying at that point, try to create a plan that is entitled or envisioning yourself in this space and makes sense for the community and the people around it. I feel like having good collaborators in the first 2 years crucial, even if you already have funding, especially in institutions that are public. It could take a while to get all the fundings like actual transfer. I will recommend having a collaborators that could help you or give you some leeway. Take your little wins because it's hard and it takes time and it's a lot of effort. In my position, I have a full teaching job and then it's the research. It's a lot of time for prepping my classes, and then I have to continue doing all research or moving forward. I've seen that other people also take a few semesters for getting lower or non-academic sabbaticals to kind of improve research in some way, take advantage of whatever those resources. I have a few discharged the first year, reducing my load of credit. It was really necessary for me being able to do my first grant and write, teach the students. Very few students, and most of the time they don't know your specific experiments or techniques. In my case, they have no experience in research before they join my lab, so I love to teach them the very basics and help them kind of like build up that love of research.
What I'm hearing is that building a community of mentorship kind of starts with yourself and being kind to yourself and patient with the process and being good collaborators, and then making sure that you provide those resources to the people that you mentor to. So the community of mentorship goes in all directions. It goes into yourself, through your collaborators, and then to the people you mentor. Can you give me an idea of what does a good collaborator do and what does a bad collaborator do?
I have two bigger collaborators. They are very different. I don't think it's actually bad, but you have to kind of ponder what is best for your interests and your needs. So I would say I have a collaborator that is providing me with a project. I teach few students basics and then put them with the graduate students that he has in the lab. And we are meeting in the middle in some ways. He has more expertise on behavior, for example, doing behavior essays and so on. And I have more experience in the microscope, analyzing data in the imaging area, or doing more tissues prep. I teach the students some aspects of the projects and kind of take over those portions of them. The other collaborator provided the resources and so on because he knew that I was still getting in. Since the tissue and the animals were already in the project, it was something that we agree on. We are working on that they have time for doing behavior analysis and have learned how to do those essays, evaluating the tissue that goes with the behavior. Another collaborator that I have, she was my thesis advisor. She kind of provided the space because there's nobody else at the moment in the lab. There's a transmission electron microscope that I can use to process some samples, and I am able to kind of use those resources. It's been great to have that available to kind of teach students to process stuff and also get some data on the side for other projects.
Going back to mentoring your undergraduates, you mentioned that for a lot of them, this is their first time ever being in a lab and doing the research. Can you share some examples of how you've been able to introduce your undergraduate students to research and help them develop their creativity and their independence in the lab?
My students, they really don't have experience, even just managing liquids, using the micropipettes and how to manage a few stuff and kind of using all these techniques that they probably learned just the theory around. So they get to practice those. And then when I have the project, I allow them to explore and think. We do journal clubs, and then I was doing a think tank or some sort of experience where they say like, okay, this is one of the ideas that I have for my grant. I want you to go and give me ideas or questions that you want to work around, and they go able to create and make a few questions, and they can look for literature and see, and then they come back and everyone discuss them and go further, and if someone has done it or not, how interesting and how it really makes the goal of our main topic or main interest in the labs. And then developing or just analyzing the images and kind of quantifying and learning. So they have developed a sense of how to keep making the right question or kind of improving it into an experiment.
You mentioned the think tank, and I think last time we talked, you were going to the Allen Institute for making neuroscience research resources for the classroom. What about these experiences make you excited, and how do you make these topics accessible to students?
In the Allen Institute, we did were a few undergraduate educators doing micro modules. The Allen Institute have a lot of data and resources. They have complete brain and all the imaging, and then they have a correlation or are able to pinpoint what specific cells and the metabolomics, proteomics. So it's a lot of data, the molecular side, imaging side, and to have a lot of ways to kind of analyze healthy and other types of tissues. So we were creating micromodules, and I hope that I can integrate this in one of my classes. We mainly focus on cell in the single cell, like organelles that they have labeled, and they have all these images that you can see the organelles through all the cell cycle. So you can kind of, for example, see the endoplasmic reticulum and different points through the cell cycle in synthesis versus the G1, and through the mitosis, how it moves, how it shapes, so they can create a sense of how different the cell is from just an image and have this view of three dimension and dynamics that is happening, and also making or having more curiosity, hopefully. In that way, they can make questions or wanting to create a way of using those data that we have available of proteomics, metabolomics, what are these changes that are occurring during different time points, and how we can learn more about this in cells that are more of cancer or muscle cell versus neuron and other types of tissues. I hope to integrate this some of my courses. We create this module specifically for these organelles to see them and the difference in shapes of cells, and then we have a few different data sets of cells that are cardiomyocytes and other single cells that are mitotic. It's been really great. I really enjoy seeing scientists and educators and how much they can kind of bring together being more accessible, and because education is a great tool, but also educating through research, it's one of those ways to bring the creativity to be able to actually expand so much more, so the students can overcome their own limits and not just these limits of content that you may have in the past of just using a textbook or a fixed data set. But if you have those big datas, and they can have so many questions, and because this is something, or the data in the Allen Institute is something that is being very recently available, there's so many opportunities for the students to do publications if they want to with this research. So they can just analyze that data and create some correlations and then publish it, and some other students in different institutions can see that and kind of continue the work around it.
That is a huge barrier to publishing that I hadn't really thought about recently, of getting data that is approved to be used for research that can be published, following the institutional review board regulations. That's so interesting and exciting. That sounds like a really cool program to be a part of.
It's a real challenge because it's such a big data. So in terms of how to download and how to actually using it in a everyday or person computer, it's been a challenge. So they are creating a few resources to kind of improve the way so you can visualize it online and don't have to download big chunks of data and trying to have all these computer science skills. So they have been more digestible, especially for biologists that may not have that skill set which is one of the challenges or the barriers. Definitely having those policies of using data that is available responsibly and healthy. There's a few donating and openly donated for these purposes of tissues that are from people that donated their brains. It's very cool.
I should probably pull up the website so we can shout out to the Allen Institute. Is this the kind of microscopy that you do?
Those are really, really great quality. I don't do that many colors, probably. I do more fluorescence, and these are a lot of 3D reconstructions. I do more confocal, a little simpler that because they have very high resolution or super resolution scopes that are really good.
I like seeing you light up when you talk about, because it sounds like you experience joy when you're mentoring students in two different ways. There's the core content that they need so that they can then apply it and really explore through research. It's just really fascinating and exciting to see the way that you light up when you talk about your students. How has your experience with the Linton Poudry leadership helped you build a network of peers that you can lean on?
It was one of those life-changing experiences where you feel so fulfilled in so many ways. Sadness is always a home for me, but this leadership institute, it's people with so many different backgrounds and so many different skill sets that you can see different ways of how to apply and connections and also impact with everyone. So yeah, to see people in science policy, to see people that are in academia in different phases or different institution types and different positions that I didn't even know, right? People that are specifically there to improve their students or faculty professional development. It was really great to see those networks and all the different topics that were really great to have that experience. I didn't get probably never in my younger years, but not at this intense or formal education. Just having those emotional intelligence topics, how to improve my communications and the ways I interact in a negotiation or how to portray myself in so many ways and kind of always thinking of your community or your impacted scenario to bring our institutions and in my case my students to the best. And it was one of those experiences that helped me to gain confidence or be more confident now that I'm in more positions of administrative labors and meetings with older and more experienced and kind of like help to shift the ideas or make sure that things move forward and we don't always do the same things, trying to have the change that is moving with the times and actually like helping improve the institution.
Our last guest was Dr. Jocelyn Bosley. She is over at the University of Nebraska Lincoln, and she had a chance to look through your LinkedIn profile and ask a question specifically for you.
I just would be interested to hear about a particularly impactful mentoring experience that she had. And it could be that it was a really good mentor that she had that made her then want to go out and be a good mentor to others, or it could be like a really negative experience that made her think, you know what, I'm not going to do that. I'm going to do this differently. But just something that a mentoring experience that impacted her.
Such a great question. Definitely one of my early days that I experienced mentorship was from a graduate student when I was in one of the summer internships as an undergraduate. She was very holistic and very fulfilled. And she will make sure that I had time to read literature, to practice my skills, but also, right, have time to go out and make sure that I have time for fulfilling my spirits and have joy because you gotta have all of those. And it was one of those experiences that allow me to kind of contrast whenever I have a mentor that was probably not allowing me to be myself, allowing me to have my bubbly personality. Then I realized like it's so much better because it's a career. So I have to do this as I love what I'm doing and not just doing it because we want to get a paper out. We love what we do. It's such a long run. And if you have the opportunity to kind of be happy and balance your life and not be all the time into the lab and also have time for sharing. So I try to incentivize that with my students during the holiday season. We do have dinner together and kind of like close up the lab in that way, like closing the year or celebrating our wins. And it's one of those times that we have for bonding and making sure. And if we travel for a conference or something, we always go. From the institute, Miguel, do you remember one of the leaders there?
Yeah, Miguel, yeah.
Yeah, he recommended one book, and I started reading it. It was called Humor Seriously, and it's one of those books that it's has data for how much just having laughter and having this levity of being willing to make a joke, even if the joke is not funny, you can have so much more connection, and it helps the students also to kind of relax, get rid of the anxiety, and learn better. So I try to make that balance. And since I've been reading this book, I realized that is something that probably in Puerto Rico we have. We kind of make fun of ourselves a lot. I try to kind of implement this better or more intentionally in classes. I have a funny story. I was at the classroom and we have a change of room, so it was not my usual. I was trying to draw some tissue so they can kind of think of extracellular matrix versus the cells and trying to pinpoint a few concepts. And I was trying to draw something, and the calibration of this board was not great. And every time I draw something, it was moving into such a different position. And it turned out like one of those Chaplin movies that you see all this comedy happening. And I was trying to get rid of the lines, and everyone started laughing so hard in the classroom, even myself.
I love that. That's so funny. Technology, sometimes it hurts more than it helps.
Yeah.
Our next guest is Dr. Stephanie Pazzo. She was also in the Linton Poudry Sagnas Leadership Institute, and you get to ask her a question. So, what question would you like to ask Stephanie?
I wonder how she's able to integrate some of the research or experiences in her classroom. I wonder if she will be able to allow the students to do a project and kind of develop some way, and how she can handle or the great leadership roles that she is in.
Thank you so much for making time to be on our podcast today. I really appreciate it.
Yeah, thank you for doing it.